Good morning, students. Now we are going to study chromosomal theory of inheritance and sex determination in the chapter of principle of inheritance and variation. First of all, coming to chromosomal theory of inheritance. This theory was proposed in year 1902 by Walter Sutton and Theodore Boveri. They noticed the behavior of chromosome and they observed that behavior of chromosome is parallel to behavior of genes. We know that Mendel worked from the year 1856 to year 1863, but his work was unrecognized. When this chromosomal theory was proposed, scientists accepted that work of Mendel was also considerable. Now, what this theory proposed? It proposed that chromosomes are carrier of genetic material. That means chromosome will take the genes from parents to offspring. Second proposal was that chromosomes are linear and genes are located on them. And segregation of chromosome will lead to segregation of genes automatically. So here in this diagram you can see this is the structure of homologous chromosomes. During meiosis 1, as we know that it is a reductional division, there is a separation of homologous chromosome, and in the meiosis 2, chromosome will split apart and they will segregate into the chromatids. And genes are located on the chromosome. So automatically genes will also segregate. Experimental proof of the chromosomal theory of inheritance was given by Thomas Hunt Morgan. He worked on fruit fly. The scientific name of fruit fly is Drosophila melanogaster. With this experiment, he confirmed that genes are located on the chromosome. He also confirmed the Mendelian law of inheritance. Apart from this, there was one more observation that certain characters are sex-linked and they will not show independent assortment. Now, Morgan chosen Drosophila melanogaster because of certain reasons, and these reasons were they could be grown on synthetic medium. That means they could be grown in laboratory very easily. Variation can be seen by light microscope. So it means no extra equipment was required to study variation. Fruit fly has only 4 pairs of chromosomes. With this, study of inheritance became very easy. Life cycle of fruit fly is of only 2 weeks. That means within a small period of time, a large number of progeny could be studied. In a single mating, large number of offsprings were produced. And more important is male and female are distinct. They could be analyzed easily with the naked eye. Because of these reasons, Morgan chosen the fruit fly as an experimental organism. Now coming to linkage and recombination. Morgan studied dihybrid cross in Drosophila and concluded that certain genes were sex-linked. So these are the various phenotypes of fruit fly. Here you can see this normal fruit fly with the white eyes, flies with the short wings, curly wings, yellow body and ebony body. So as we studied earlier that certain traits could be analyzed without help of special microscope. While studying this, Morgan observed when yellow-bodied and white-eyed males are crossed with brown-bodied and red-eyed females, then in the progeny, parental combinations are higher in number and recombinants are very less. So this ratio varied considerably from the dihybrid cross of Mendel, that is 9:3 is to 3 is to 1. So Morgan concluded that certain genes they try to remain together. Linkage. The density of DNA sequences that are close together on chromosome to be inherited together during meiosis is called as linkage. This lowers the chance of recombination. And recombination is generation of non-parental combination in the offspring. So let us understand what is the genetics behind this. So here in this diagram, you can see that there are two genes. First is capital G and another is capital A. As these genes are located far apart, that's why during the crossing over, there are chances of moving from one chromatid to the another chromatid. So here in this, we are going to found a non-parental combinations also. While in this diagram, you are seeing that both the genes are located very closely. So during the crossing over also, they are moving together. So these kinds of genes are linked. And here you can see that generation of non-parental combination is either absent or very less. Now coming to sex determination. Sex can be determined in various ways like X and XY combination, XX and XO combination, ZZ and ZW combination, and with the help of autosomes. Now let us study all these one by one. First of all, coming to XX and XY type. In this, father is having the 6 chromosomes X and Y, while mother is having 2 X chromosomes. Now coming to the offspring. Generation of gametes. So here you can see these are the gametes which have been generated from the males and these are the gametes which have been generated from the females. So this is a progeny. Now result. Chances of male is to female is equal. That is, there are 50% of chances for male being born and 50% of chances of female being born. Here the female is homozygous while male is heterozygous and gender of the child is determined by father. This kind of pattern is observed in we human beings. Now coming to XX and XO type. Here the father is having XO, that is single sex chromosome, that is X, and Y is absent while female is showing 2X chromosomes. Now generation of the gametes, male and female gametes, making of progeny. So here we also see that making of male and female is in equal ratio. Here the sex of a child is determined by the male, as males are heterozygous while females are homozygous. Now coming to another kind of sex determination, that is ZZ and ZW type. Here, the father is homozygous, while mother is heterozygous. Now, making of male and female gametes, generation of progeny, coming to the results. Here, the females are responsible for determining the gender of a child, as female is heterozygous while males are homozygous. This kind of situation is found in birds. Now coming to another type of sex determination, that is with the help of autosome, or we can say that haploid and diploid type. In honeybee, females are diploid while males are haploid. So here see in this diagram, when female bee is showing meiosis, it will make eggs which will be haploid, and with the no fertilization they will mature into male bees. When these eggs fuses with the sperm, then there will be the generation of female bee. When the female bee will feed on royal jelly, then it will change into queen. Now understand this line. Males do not have father, but they have grandfather. That means here you can see in this, the male, they are being born by females only. So they don't have the father. But for the birth of females, males are responsible. So we conclude that males don't have father, but they have grandfather. Now coming to the assignment. These assignments will make you understand the content better. Thank you so much.